When we imagine a cell, it's tempting to picture it as a tiny bag filled with chemicals. But that picture couldn't be more misleading. A living cell is a regulated system separated from its environment by a membrane that is both protective and selective. Nothing meaningful happens unless that boundary is respected. The question this video answers is simple but profound. How do cells move materials in and out without tearing themselves apart? The answer lies in two coordinated processes, endocytosis and exocytosis, which allow cells to import nutrients, export signals, and maintain balance, all while keeping their membrane intact. This is not passive movement, this is intentional cellular logistics. Here we see the challenge every cell faces. A cell exists in a crowded environment filled with nutrient-signaling molecules, waste products, and potential threats. Yet the cell membrane is selectively permeable. That means many substances, especially large or charged molecules, cannot simply diffuse across. Glucose, proteins, neurotransmitters, and waste are all essential, but they're too large or too polar to pass freely through the lipid bilayer. If diffusion were the only option, cells would quickly lose internal balance, what we call homeostasis. So transport systems exist for one core reason, to allow controlled exchange while preserving internal stability. Without these systems, cells couldn't regulate energy, communicate with neighbors, or survive longer than minutes. This slide introduces endocytosis, the process by which cells bring material into themselves. Visually notice what's happening. The cell membrane begins to bend inward, wrapping around external material. Eventually, that membrane pinches off, forming a vesicle inside the cell. That vesicle contains whatever cargo the cell needed, nutrients, particles, or signaling molecules. This process is active, not passive. It requires energy, coordination, and precise membrane remodeling. Endocytosis tells us something important about cells. The membrane is not rigid, it's flexible, responsive, and under tight control. Cells don't open holes in their boundary, they reshape it. Not all endocytosis is the same. Cells use different strategies depending on what they need. On the left, we see phagocytosis, often called cell eating. This involves engulfing large particles such as bacteria or cellular debris. Immune cells like macrophages use this process to protect the body. In the center is pinocytosis, or cell drinking. Here, cells non-selectively take in extracellular fluid and dissolved substances. It's bulk intake, less specific but constant. On the right is receptor-mediated endocytosis, the most selective form. Specific molecules bind to receptors on the membrane, triggering vesicle formation only when the correct ligand is present. This distinction matters because it shows that cells prioritize what enters. Not everything gets equal access. This is cellular decision-making at a molecular level. Now we shift direction, from import to export. Exocytosis is how cells send materials out into their environment. Inside the cell, molecules such as proteins, hormones, or neurotransmitters are packaged into vesicles. These vesicles travel toward the membrane, fuse with it, and release their contents outside. Notice the symmetry here. Endocytosis brings materials in by membrane fusion. Exocytosis sends material out by membrane fusion. The membrane remains intact the entire time. This process is essential for cell communications. Neurons release transmitters this way. Endocrine cells release hormones this way. Even digestive enzymes are secreted via exocytosis. Without exocytosis, multicellular life would be impossible. This slide emphasizes a critical idea. Transport is intentional, not automatic. Both endocytosis and exocytosis require ATP, the cell's energy currency. Vesicles don't drift randomly. They're actively moved along the cytoskeleton, guided to precise locations. We also see two forms of exocytosis: continuous exocytosis, which happens constantly, and regulated exocytosis, which occurs only in response to specific signals. Timing is everything. For example, In neurons, neurotransmitters must be released at exactly the right millisecond. In pancreatic cells, insulin release must match blood glucose levels. When this timing breaks down, disease follows, such as diabetes, neurological disorders, immune dysfunction. This reinforces a key theme. Cellular transport is not about movement, it's about coordination. Here we zoom out, from the molecular level to the scale of life itself. At the cellular level, transport maintains balance. At the tissue level, it enables coordination. At the organism level, it supports identity function and survival. Neurons communicate by releasing neurotransmitters. Immune cells protect us by engulfing pathogens. Hormonal systems regulate growth, stress, and metabolism. Every thought you think, every immune response you mount, every heartbeat you feel depends on these transport systems functioning properly. When they fail, cells lose balance. When balance is lost, disease emerges. So endocytosis and exocytosis are not just cell biology topics. They're foundations of health. Thank you for watching, and if you want more, subscribe to EQBloom.